So hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Shuyuan Yao from WuXi Advanced Therapies.
Hello, everyone. Today, I will present our high-quality plasmid DNA production platform. First, I would introduce WuXi ATU. WuXi Advanced Therapy have 7 sites across the globally and in 2 countries. In United States, in Philadelphia, we have 400,000 square feet and working on viral vector manufacturing, cell therapy manufacturing, and testing. And in China, we have 380,000 square feet doing viral vector cell therapy and testing. And we also have R&D center in China. And with these 2 sites, totally about 900 people, we provide full service on cell and gene therapy to accelerate your cell therapy product development. Here is the U.S. site. You can see in U.S. side in the Philadelphia, we have 4 buildings, Legal Island 1, Legal Island 2, and C3 and R400. Legal Island 1 and 2 are for cell and gene therapy manufacturing. Legal Island 1 is for clinical manufacturing. Legal Island 2 is for more for commercial. For CC3 building is for cell therapy, and for R400 is for testing. In China, we also have 4 sites. 1 site in WuXi City in Jiangsu Province, and the other 3 is all in Shanghai City. 1 is in Waigaojiao, another one is in Lingang, and the third one is in Jinsao. Jinsao provides oncolytic virus service, and the main campus is in Jiangsu Province, WuXi City, and the platform we're introducing today for Plasmid service is in WuXi City. As a global CMO, CDMO provider, we provide high quality and regulatory compliant service to all the clients in all over the world. And here is some record of our inspection record from either our audience or the agency. So here is in China, we provide the service platform we provide, including the end-to-end PD service and plasmid and viral vector and cell therapy. And as I said, in Jinsao, we provide oncolytic virus production and research development service. And for the we also provide quality control and assay development service. So in the next section, we'll talk about the plasmid in cell and the gene therapy, their application, and also what kind of requirement they need for different application. Here it shows the 3 major applications for plasmid in cell and gene therapy. The first one is in vitro application. We're using plasmid DNA to make viral vectors, such as lentiviral vectors and AAV vectors. The second application is ex vivo cell therapy, such as making CAR T cells with DNA plasmid, and also with making iPSC cells with DNA plasmid. And the third application is in vivo gene therapy, including DNA vaccine and the DNA templates for gene editing. For different stage of the drug development, you need a different grade of plasmid. For for the early development, such as discovery and development, you may need a research-grade plasmid. And for the development and early phase, for example, phase 1, you may using GMP-like grade plasmid. And for the clinical manufacturing and the commercial manufacturing, you definitely need a GMP-grade plasmids. You can see there are some green areas. It's really the customer's choice. You can either using either one. They all work, depends on your strategy of your development. So plasmid is a very common item for the biology major people. But when we using plasmid for cell and gene therapy product, there are still some challenges. The first challenge is the scale. So at different development stage, you need a different scale for plasmid. So the key here is you need to keep the consistency, the process consistency from beginning until the end of the development. And another 3 challenge is the timing. So because the short of a provider, so the plasmid service of the plasmid, the DNA plasmid always take a long time, lead time, and wait for the clients to wait. For the cost is one of the thing is always sensitive for the developer, especially when we're using large scale DNA for lentiviral or AAV viral packaging. So the cost is always a sensitive issue. The quality. So you need to make sure your plasmid quality can meet your country's requirements. You know, also when you do multi-country, multi-center, multinational centers, so you need to using different quality for different country. For next one, we talk about the WuXi ATO plasmid platform. For manufacturing of plasmid, they're including 2 parts. The first part is bacterial banking. The second one is plasmid manufacturing. The plasmid manufacturers come from the amplify of the bacteria holding the plasmid. So the first step is build up the bacterial bank from the research cell bank to master cell bank to working cell bank. This take about 14 days for the production and plus 45 days of the quality control testing. For the plasmid manufacturing, after the fermentation, you need to harvest the bacteria and then lysate and purify it. The whole process take about 6 days. In our layout, we can do 3 days per lot, and I will give the detail in next slides. The QC takes about 1 month. Here's the detail of the cell banking of the bacteria. And you can see at the very right is the layout of the bacterial banking facility we have. And in the middle is the process. And here for this facility, we have 4,000 square feet and ISO 7 cleanliness areas. And here is the key here is the enclosed system and single-use consumables. And we think this is the only way to avoid cross-contamination from different batches. So we're using like a enclosed system. So this slide is for plasmid bacterial banking testing platforms. For the testing, we have 3 aspects. One is the strain identity, and the other one is plasmid-related characterization, and the third one is plasmid identification. So you can see at the right, we have different testing criteria for RCB, MCB, and WCB. In the middle is the working days for each of the testing. You can see the longest is 45 days. At the different stage of your product development, you need a different scale of the plasmid. At the early stage, you may want to need a small scale, and at the late stage, you want something like a higher, large scale. And we do that in 2 ways. The first one is 5-liter bioreactor. We can parallelize them, and we can parallelize 5 of them to make a working volume of 3 to 16 liter, and in this way we can provide 25 to 500 milligram. And the larger one is 200 liter bioreactor. We can provide working volume 30 to 150 liter, and to provide 500 to 5,000 milligram, which is 5 grams. So for some case, we can provide 10 grams. Here is the plasmid manufacturing platform. So and you can see again at the very right is the layout of the facility. You can see in this facility we have 8,000 square feet, and we have ISO 8, ISO 7, and ISO 5 clean areas, and depends on the different step of the purification. And again, here the design is single direction personnel and the material flow, and we're using enclosed system and single-use consumables to avoid possible cross-contamination and easy changeover. And again, the scale is from 25 microgram to 5 microgram, and we can do 3 days per lot, because the design of our facility, we can do the fermentation and then pass through to the purification, and then we can start the second batch of the fermentation. In this, we can do 3 batch per lot and 100 lots every year. And here is the Plasmid product testing platform. This is a product release testing. You can see for PD run, engineering run, and the GMP run, we have different kind of criteria. And again, in the middle is the testing days needed. And again, the longest is 28 days for the sterility. And so therefore, the whole releasing time is about 28 days. Here's next slide. We talk about the advantage of our WuXi ATU plasmid platform. And in our platform, again, we provide different manufacturing scale to meet your different stage, development stage, and quality need, quantity need. And we have like a much quick turnaround time, and depends on the the starting material. We can finish your plasmid manufacturing in 2 to 3 months. And again, we have reduced the cost, and we have the high quality, and our regulatory support can support in Europe, in China, and in US. Here is the current project we are going, and you can see we have 3 projects, R&D projects for clients from the US, and we have 3 R&D projects ongoing for clients from UK, in Europe, and in Korea we have 4 clients, and in China we have 8 clients. And actually in China we already finished one of the clients R&D in last year. So here is all the projects we are going in our China ATU. This including Plasmid and AAV lentil viral vector and oncolytic viral vector. You can see our service covers different stage, and including IND enabling, IND approval, and BLA enabling. And we have a quite broad service for all the clients all over the globally. That will be all for today. Thank you.
Okay, great. Thanks. So the first question is, if your plasmids are manufactured in China, are there any challenges in import or export to anywhere in the world?
In general, there's no problem, but because plasmid is a bacterial material, so you need to get import export permit for the customer clearance. So WuXi ATU will provide service to for all this application, and the lead time is about 4 to 6 weeks, and then we can start earlier with, so that we don't delay any of your service. And we also provide de novo synthesis for the plasmid. In that case, you can simply give us the information. We can synthesize DNA plasmid for you, and we don't need for waiting for the importation permit.
Okay. And what is your current availability and turnaround time?
So currently, we have no more than 2 months lead time for your plasmid production, and the turnaround time is about 3 to 4 months for the whole service period.
Okay. And what is your scale for GMP-like and GMP-grade plasma?
For the GMP-like and the GMP-grade plasmid, there's no scale different. So it's all about 200 microgram to 5 microgram in the small scale. For the large scale, we can do up to 5 or 10 gram.
Okay. Well, thank you, Shuyuan.
Thank you.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.